Just like Jeremy did, I just like to iterate that the importance and the impact from IDG truly stems from the outcome of all of the IDG members, which range from the data resource generating centers to the knowledge management center to the cutting edge informatic tools awardees, working together with the resource dissemination and outreach center for facilitating and coordinating the outreach and exposure of these various outcomes from IDG. I would also like to note that outside of the IDG consortium, NIH offered additional druggable genome support in order to enhance research into these understudied areas. Those included multiple rounds of IDG-associated R03 grants, administrative supplements for the HEAL initiative, looking into associations between pain and IDG-eligible targets, as well as commercialization support via SBIR and STTR grants. Next slide, please. In the 9+ years that IDG existence, there have been over 500 publications from these IDG consortium grants, and these have elicited over 51,000 citations, which are still growing in magnitude and in impact factor. Furthermore, a collaboration was set up with the editors of the Nature Reviews drug discovery manuscripts for establishing the IDG Target Watch in 2019. These summary reports highlighted new findings for understudied targets, and in total, there were 22 manuscripts in this series with over 150 citations so far. There are even citations in the current year, 2024, of IDG target watches published in 2019, indicating that these early IDG work impact further work into these understudied targets. Additionally, part of this series included an annual publication of the analysis of novel drug targets for newly approved drugs from the FDA and other world agencies. These analyses are a part of the Drug Central database, which you'll hear later about, and are a part of the elevation of a target into this TCLIN classification that Jeremy just explained. Next slide, please. And to expand the exposure of IDG, we also established the IDG Webinar Series with presentations from IDG members and invited speakers. And over the 5 semesters of these webinar series, we had consistent attendance from all over the world. I would like to especially point out a speaker during the 2023 series, Dr. Nicoletta Landsberger, a professor from the University of Milan. She was a recipient of an Italian Research Foundation grant. The grant call was based on researchers utilizing TDL, created by KMC, for defining understudied targets, such as TDART, and finding the connections of these TDART targets with rare diseases, indicating the impact of KMC work outside of the United States. Next slide, please. Additional IDG outreach events were organized with other groups with the hope of enhancing collaborations and extending research into these understudied areas. So IDG held meetings with a number of different public-private partnerships, which has members from academic institutions, biotech and pharmaceutical industries. And these meetings included meetings with Open Targets, ATOM, which is the Accelerated Therapeutics for Opportunities in Medicine, and the Target 2035, which is an extension of the Structural Genomics Consortium. We also hosted invited speakers from the EUB Open and IMI, which is the Innovative Medicines Initiative. As well, there were 2 IDG-centric FACET meetings organized by IDG members. In total, these events were attended by over 2,500 people and strengthened the resolve to investigate these understudied areas. Next slide, please. Another venue, just like is done here, is that IDG influenced and had outreach is via our YouTube channel, the Druggable Genome IDG. It was here that we posted recordings of presentations and demonstrations from various events. And to date, we have over 300 hours watched by 4,500 viewers, and the subscribers continue to increase, indicating a continual IDG interest. Next slide, please. Before I let you hear more about the data resources generated by IDG, I'd like to point out the IDG reagent resources and their impact to date. These are resources for proteins on the IDG target list only, namely that subset list of the 3 druggable protein families, the kinases, GPCRs, and ion channels. Annotations of these resources and data types were submitted into the resource management system set up by the University of Miami. And this graph depicts the magnitude of the resources based on type. To date, there were almost 1,000 records with close to 400 with complete annotations, which we deem FAROS ready and are shown in FAROS. Many resources have also been deposited with commercial vendors such as Addgene and Millipore Sigma for ease of distribution. Furthermore, IDG was an early adapter of the research resource identifiers, those RRIDs. Hence, we can now track publications utilizing these IDG resources via SiteCrunch. And to date, there are almost 300 publications mentioning IDG resources and/or projects. And we hope that continues to increase. With that, I thank you for your attention, and I pass it back to Jeremy.